Methods of propagation.
The first one is the seeds or the sexual propagation.
It includes seed collection, seed testing, seed storage and handling, and seed germination. There are three ways to collect seeds from selected mother trees: the collection from the ground, collection from standing trees, and collection by shaking branches. Seed processing. We separate the seeds from impurities through flotation. Fill a container with water and submerge the seeds. Seeds that float are empty, therefore will not germinate. Seed testing is done to determine the quality and capability of seeds to germinate into healthy seedlings. Seed storage is a technique where seeds are kept under favorable environment conditions to maintain seed viability.
When the seed starts to sprout, germination is taking place. Sterilize the soil before using it as potting material to avoid problems with soil-borne diseases, such as dumping off and root rot. This is done using any of the following methods. Heat the soil over a fire for about 15 minutes. Apply chemical disinfectant. Pour boiling water or spread the soil under direct sunlight in a clean area for 1 or 2 days. Collect wildlings when the soil is moist to minimize damage of roots. Gather the wildlings by simply lifting them with the aid of a bolo or shovel. Leave a ball of earth attached firmly around the roots. Plant the wildling on a plastic bag with hose. Transfer the bold wildlings to a shaded portion of the nursery. Let them develop before planting in the field.
Asexual propagation is a method of plant reproduction that does not involve the union seed. New plants are produced from the parts of existing plants, such as leaves, roots, stem, and buds. Asexual propagation involves cutting, layering, division, and grafting, among others.
Our first step is selecting your cutting material. It's crucial to choose a healthy parent plant from which to take your cutting. Look for a young but mature stem or branch that's not too woody or too green. This ideal cutting material will ensure successful propagation. Once you've selected your cutting material, it's time to prepare for the cutting process. Equip yourself with a clean, sharp knife or scissors and cut a piece of the stem about 4 to 6 inches long. Make sure your cutting includes at least 2 or more sets of leaves or nodes. For rooting, you can collect young shoots or cuttings and trim off their leaves into half. Place them in a container with water or cut them into 2 nodal cuttings. Don't forget to scrape the basal portion of the cutting for optimal root development. Prepare a pot with well-draining soil. Choose a pot that just slightly larger than the cutting to provide ample room for root growth. Next, it's time to insert the cut end of the stem into the hole in the soil. Be sure to make the hole deep enough to accommodate the entire length of the cutting, but not too deep that the leaves or nodes are buried. Gently but firmly press the soil around the base of the cutting to provide support and stability. To ensure the survival of your cutting, creating a humid environment is essential. You can achieve this by covering the pot with a clear plastic bag or placing it in a mini greenhouse. However, it's crucial to ensure there's enough room inside so that the plastic does not touch the leaves, as direct contact can promote fungal growth. After a few weeks of care and patience, it's time to check for root development. Gently tug on the cutting to feel if roots have formed and are resisting the pull. Once rooted, it's time to lift the cuttings and dip them in water to gently remove the soil from the roots, preparing them for the next stage. The final step in our cutting process is transferring the potted cuttings to a hardening area. After 4 to 8 months of growth and development, your cuttings are now ready for outdoor planting, where they'll continue to thrive and flourish.
Grafting. Connecting two pieces of living tissues together so that they unite and eventually grow and develop as one plant is called grafting. This propagation method is used when cutting is not applicable. It is also used to hasten reproductive maturity of seedlings, establish clonal seeds, orchards, clonal tests, and clonal banks, obtain benefits of rootstocks. There are several types of grafting such as cleft, saddle, splice, and whip or tongue, among other methods with respective procedures. Following steps are generally used in grafting. Remove the leaves but retain the buds or the nodes in the scion. Split cut the top end of the selected stalk. Sharpen the base of the scion. Insert the scion between the split cut of the stalk. Apply wax over the cut portion to prevent loss of water. Tightly wrap the grafted part with a plastic sheet. Do not cover the nodes. Cover the grafted portion loosely with a plastic bag. Remove the plastic bag with young leaves appear but leave the plastic sheet. Remove the plastic wrapping after 5 months. Cut the grafted portion and plant the graft. Marketing. This is a propagation method which includes the development of roots on a stem while it is still attached to the parent plant. The procedures for these methods. First, select a branch that is 0.5 to 3 cm in diameter. Second, remove 3 cm of bark around the branch. The exposed part must be 15 to 30 cm from the tip of the branch. Clean thoroughly the exposed portion with a knife. Cover the exposed portion with moist rooting medium such as sphagnum moss or coconut husk with small amount of rich, loose soil. Fifth, cover the rooting medium with plastic. Tie both ends with string to prevent seeping of too much water. Cut the marketed branch below the wrap portion when the roots are fully developed. Lastly, 7. Remove the plastic and plant the market. Keep the soil moist.
Budding. Budding is done when the scion is reduced in size to contain only one bud and a small portion of the bark, with or without woods. This process is applied or used in small plants. A scion is the detached living portion of a plant, such as a bud, to be attached to a rootstock in the budding or grafting methods. There are two types of budding. One, shield budding. This is widely used in citrus.
Make a horizontal cut in the rootstock, 2 to 3 cm long as deep as the bark.
Make a vertical cut below the horizontal cut, forming a T. Slice a shield of bud from the bud stick about 2.5 cm under the bud to 2 cm above. Number 4. Open the bark in the T-shaped cut in the rootstock. 5. Insert the shield by pushing it downwards under the 2 flaps of bark. 6. Tie securely with a plastic tape but leave the bud exposed. Don't press the tape too firmly against the inserted bud. This will destroy the growing bud. Patch padding. This is widely used on fruit trees with thick bark. Number 1. Remove a rectangular piece of bark from a rootstock. 2. Slice a rectangular patch containing the bud from the bud stick. 3. Insert the patch with the bud in the cut on the rootstock. The inserted patch should fit the cut tightly. 4. Wrap the part with the inserted bud with plastic, tape, or wax cloth, leaving the bud exposed. 5. Trim the upper portion of the rootstock just above the patched bud. 6. Cover the stem portion of the grafted bud with a clear plastic bag to reduce transpiration. 7. Water the rootstock daily. 8. Remove the plastic cover once the grafted buds sprout. Tissue culture. Tissue culture is an alternative means of propagating superior planting materials in a sterilized laboratory. Using a piece of a plant part grown either in a test tube, bottle, or other containers, they are provided with appropriate rooting media under controlled conditions of light and temperature. Tissue culture can produce a larger number of quality seedlings. This method, however, is more expensive. To venture into tissue culture, these are the following steps. Number 1. Select a mother tree in the natural forest or in the plantations. 2. Collect cuttings or marcot branches from the mother tree, which will be source of young shoots. 3. Sterilize young shoots using disinfectants. 4. Inoculate the explant into culture medium, agar. The culture medium will supply the nutrients needed by the plants to grow and develop in the test tubes or bottles. 5. Induce the explants to form multiple shoots by using growth hormones. 6. Provide cultures in the laboratory with lights and low temperature. Acclimatize the microshoots with nursery conditions while growing inside the bottles. 7. Upon reaching 1.5 to 2 inches height, outplant the microshoots in the trays that contain sand or soil. Enclose the tray in a plastic bag in order to maintain high humidity and low temperature. Once rooted, the plants can be exposed to normal growing conditions like any kind of seedlings.